I'm going to give you an update on where we stand clinically and with some of the newer treatments as well. Okay, so I never, even though there's just a small group so far this morning, I never pass on an opportunity to say in front of any size audience, just reminding us that we need to be cognizant of some of the ways that we conceptualize lung cancer. And in a couple days, it's going to be November, which is Lung Cancer Awareness. So I think one of the major obstacles we have is this notion out there in the public that this is the only guy who can get lung cancer. Essentially, if you have lungs, you can get lung cancer, and the face of lung cancer really is changing. And here's some pictures of some of our patients at MGH, and you can see the resemblance to the last slide is not necessarily there. And it's because of this thought that people have that only someone else can get lung cancer. I think that we see a lot of funding inequities. So the size of the person-shaped logo here is the number of deaths that, this slide is a couple years old now, but the number of deaths that we have annually from each different type of cancer: lung, colorectal, breast, and prostate. And then next to it, you can see the dollar sign, which shows the relative amount of funding, governmental funding that goes towards each of these cancers. So anyway, I think increased understanding that the face of lung cancer is changing will help bring more research into this, research funding into this field. But even despite that unequal playing field, as it were, there have been great advances in lung cancer research in the last 10 years. And we really now have entered this era of genotype-directed therapy, which was a conceptual idea 10 years ago and is really the way we practice in clinic every day now, where we test patients that all come in with seemingly the same diagnosis, for example, adenocarcinoma, and instead of treating them all with the same algorithm of different chemotherapies, we can really parse them into different groups that will have different treatment algorithms based on their driver oncogene. And each year this pie seems to have more and more pieces added that tell us the driver genotype that may correspond to an active treatment. Some of these pieces of the pie are quite small, 1 or 2%, but because of the magnitude of lung cancer, this is still even a 1 or 2% of lung cancer is 20,000 person disease. And what I always tell the fellows is that really, in order to make these targeted therapies work, you need two components. So the analogy here is you have something valuable that you've put in your safety deposit box, and in order to get it out, you have to follow two steps. One is to open this big, complicated vault door with lots of levers and different moving pieces, but then you also have to have the specific key that goes into your specific lockbox. And so the analogy for targeted therapy is that unlocking the vault door is understanding the biological context of a certain cancer, really understanding what makes it go and what its certain susceptibilities are. But even understanding that biology is not enough to treat patients. You really need to have a drug. That's the key to the lockbox. You have to have a drug that's specific for that context, that's safe for people to take and tolerable. Otherwise, you won't have the successful treatments. And this slide I borrowed from Alice Shaw, so thank you, Alice. I'll be building on this later down the talk, but essentially when you have a sensitive oncogene-addicted cancer, the idea is that the majority, if not all, of the significant downstream pathways are really driven by signaling from that receptor. And so it's because of that sort of unique dependence on the signaling RTK that we're able to block it with a specific drug and really shut down all the signaling within the cell. And in patients, this translates into these great responses. So this happens to be an EGFR patient getting an EGFR TKI, gefitinib. But as we've seen, this can translate into really any type of oncogene-addicted lung cancer when you have a specific drug that blocks that pathway. So just to go back historically and tell you a little bit about EGFR and about MGH's role in the history of this, this story. Around 2000, two different companies came out with an EGFR inhibitor. One was gefitinib, one was erlotinib. These were both oral TKIs that blocked very specifically EGFR. And so clinically what we did was what we did with any new agent at that time. We basically added it to standard of care chemotherapy to see if it improved outcome. And so these 4 Kaplan-Meier curves represent 4 large clinical trials that were done around the year 2000, where a platinum-based doublet chemotherapy, which was a standard first-line treatment for lung cancer, and in some cases still is, was given with either placebo or the EGFR inhibitor. And you can see that unfortunately all 4 of these large studies were densely negative. But even in the early studies, there was some indication that there was a subgroup of patients who seemed to do well with this treatment. So if we look at subgroup analysis from one of those 4 randomized studies, the Tribute study, which was carboplatin paclitaxel with or without erlotinib, we can see that they had thought ahead to look at the never smokers, because in earlier stage studies with this drug, they had seen some amazing responses in never smokers. And this is, as we now understand, somewhat of a surrogate for having an EGFR mutation. So when they looked at the patients who got chemotherapy with erlotinib or with placebo, who were never smokers, they essentially saw a doubling of the survival. And this was done in an era where you could not get EGFR TKIs outside of the setting of a trial. So the group here in the blue line, the placebo group, those patients most likely never had an EGFR TKI during the life of the course of their disease. So this is an experiment that we can never go back and redo now prospectively, but there's a suggestion that there's not only a response benefit, but quite a bit of survival advantage to treating these patients with an EGFR TKI. We'll come back to that in a few slides. So this was kind of the context. It seemed like maybe there was a group of patients who benefited, but it wasn't until this article ran in the Boston Globe that we really sort of understood which patients were going to be the big winners here. So this is a story that ran in the Sunday Globe in 2003, now really 10 years ago, about a patient named Kate Robbins, who was then a patient of Tom Lynch, all of our mentors here at MGH in the thoracic group. And Kate is now a patient of mine. She's still doing very well. So this was kind of a public interest story about this young woman, healthy, never smoking, had young kids in elementary school who had been stricken with metastatic lung cancer, and it was very advanced. Chemotherapy wasn't working. She had brain mets, she had liver mets, and she basically knew she was going to die soon. So in this story, it's talking about how she's writing out all these this death journal that was meant to guide her children through important events in their life that she was going to be absent from. And essentially what happened is Tom put her on, as a last-ditch effort, on this open access protocol for gefitinib. And she had this Lazarus rise from the grave type of response, and they wrote a story about it in the Globe, saying how isn't this cool, if only we understood why it worked for her and not some other people. And Dan Haber was at home on a Sunday reading the Globe and saw this story about a patient treated at his cancer center, and decided that we have to figure out what's in her tumor that's causing her to have such a specific response to this drug. So Monday morning, he calls up Tom. They got to work trying to figure out what was going on in Kate's tumor, as well as some other patients who had had similarly remarkable responses. And within 4 months, a New England Journal paper was published describing EGFR mutations. And now we know that we can test for these prospectively and try and identify patients who will benefit. As a side note, Kate is still on gefitinib. She's still doing very well. Her son is graduated from college and is working. Her daughter's going to be graduating this year. So, this made an enormous difference in the life of her family and her. And unfortunately, it is kind of a rare outcome for someone to be on one of these targeted drugs for a decade. But one of the things that we need to figure out is how to turn more patients into this type of story, to have a really prolonged outcome response to these drugs. So essentially where we stand now is that these drugs are standard first-line treatment for patients with EGFR mutations. And that is based on a series of randomized trials. I'll show you a couple slides on IPASS, which was the first randomized trial. So again, people knew that these drugs worked in patients with EGFR mutations, but they didn't really know if they improved outcomes compared to chemotherapy. They thought that potentially EGFR mutant patients just had a better, more indolent disease and they would do well no matter what you treated them with. So this randomized trial was done in Asia, where for reasons we still don't understand, EGFR mutations are 3 to 4 times more common than they are in Western populations. And patients were randomized to receive first-line gefitinib or first-line chemotherapy. And the other thing that I'll point out is that, in the US, the majority of oncologists, when the EGFR story came out, they weren't so keen on testing patients for EGFR. They thought that, well, since EGFR mutations seem to happen more often in never smokers, in women, in Asian patients, I'll just decide based on those clinical characteristics who to give these drugs to, because the genetic testing does take time and money and tissue that was in larger quantity than an FNA can provide. So this study was instrumental, I think, in helping us understand the importance of genotype screening, because the results of this study basically showed that the progression-free survival, which was the primary outcome, if you look at the box there, the hazard ratio, you can see that it's quite a positive study. 0.74 with a p-value having lots of zeros in the front of it. But when you look at the curves, it doesn't look like a grossly positive study. They cross. Essentially what this means is that there are 2 different populations within the study cohort that acted in essentially opposite ways, and we can see that bear out when we look at the subgroup of patients that had tissue available. So this study was, you did not have to have an EGFR mutation to get in. You just had to be a never smoking Asian patient with adenocarcinoma. And so this single slide really changed practice in the US, because, like I said, oncologists were slow to buy into the fact that they needed to do genetic testing. If you look at the left-hand side of this slide, you see that among those who were mutation positive, there was even a larger benefit to first-line targeted therapy with a hazard ratio of 0.48. But what made a difference in clinical practice was the right-hand side of the slide, where those patients who were never smoking, adenocarcinoma, Asian patients, if they did not have the genotype, the EGFR mutation, they did very poorly with gefitinib. So after this trial came out and was published in the New England Journal in 2009, that's really when I would say the majority of practicing oncologists started testing patients for EGFR mutations. And now we have a series of 8, I believe it's up to 8 positive randomized trials, essentially looking at this same type of design. Most of these prospectively selected patients at this point for EGFR mutation positivity, and randomize them to either gefitinib, erlotinib, or afatinib, any of the 3 approved EGFR drugs, versus platinum doublet chemotherapy. In different trials, some of them were taxane-based, some of them were gemcitabine. The Lux-Lung-3 trial had pemetrexed, but the results are very similar across the whole slide, where the response rate is 2 to 3 times higher for the genotype-specific therapy, and the PFS, which is the primary outcome for all of these trials, is markedly positive with hazard ratios that range from 0.16 to 0.58. Now, the tricky thing is that the overall survival does not seem to be improved. Some of the studies have a trend one way or the other, but for the most part, overall survival seems to be equal. Now, this, I think, is because once you know these patients have an EGFR mutation, if in the study they're randomized to chemotherapy, if they progress on chemotherapy, they're, for the most part, all going to get an EGFR TKI. One or another of these drugs is available in really every corner of the world. So I had mentioned earlier that we will never repeat this type of study prospectively at this point, where we have patients with EGFR mutations that never get an EGFR TKI. But I do believe that it would improve survival, as is hinted at by this analysis from 2005. So before turning to some of the acquired resistance, I just want to say the other thing that's really changed in the clinic in the last couple of years is this notion that when patients with an EGFR mutation progress on an EGFR TKI, that we don't take that EGFR TKI away. we tend to now keep the TKI going and add chemotherapy onto it. And here's an example of how well it can work. This is a patient of mine. You can follow the sequence across the slide. From the time of diagnosis, we found that she had an EGFR mutation. She was treated with first-line erlotinib and had a very good response. But as is common, about 15 months later, she had acquired resistance. And then we put her on chemotherapy with the TKI, and she had another great response. And just last month, or maybe 2 months ago, in the Oncologist, Sarah Goldberg, a former fellow of ours, published a retrospective series showing just this illustration that the response rate is much higher in these patients. There are ongoing prospective trials, both in the US and Asia, looking at this. And the reason for doing this essentially is that there's this phenomena of disease flare upon discontinuation of an EGFR TKI, even in someone who is progressing through their therapy. This was first published from the Memorial Group, but has been widely clinically observed and really recognized as an important phenomena since then. So in this publication, they defined a flare as actually being so sick that you needed to be hospitalized or that you died. So these are pretty significant downturns in patients. They either got hospitalized or they died. And this happened in 25% of patients whom you discontinued an EGFR TKI in the setting of radiographic progression. And the median time that this occurred was 8 days. And so the thought here is that even though you have some resistant tumor that's progressing radiographically, that's just the tip of the iceberg. There may be a lot more cancer that's actually still sensitive to the TKI, and if you remove that suppression, all of the cancer can grow. So this has also become a very important clinical paradigm. So two major obstacles remain, or at least two, in our current treatment of EGFR patients. One is that not all patients will have an initial response. So, most of these agents have a beautiful looking waterfall plot. This happens to be a study of first or second line afatinib in patients with an EGFR mutation. And you can see that all of the patients in red, so the majority, I believe it was 65% in this particular publication, have a RECIST response. So that's tumor shrinkage that meets a certain threshold, the dotted line here. But the blue portion of the curve is patients who, many of them have a shrinkage, but not necessarily a RECIST response. So Jeff's lab actually has been doing a lot of work looking at what are the modulators of response and how come some patients have a very deep response and other patients have a more shallow response. If he has time, he'll cover some of that in his talk. And our second problem, I think, is that the responses that we do see are often short-lived. So here are 3 example studies, randomized studies with gefitinib, erlotinib, and afatinib. And you can see the majority of patients are not like my friend Kate Robbins. They don't get a decade of response. They have on average 9 to 13 months. And I include this picture. It's not lung cancer, it's melanoma, but to me it's just such a graphic image of acquired drug resistance. So this is also a paper published by one of our fellows, Nick Wagley. This is a melanoma, BRAF mutant melanoma patient who was treated, I believe, with vemurafenib, who, their disease is so visibly evident in column A, and then column B is them on the targeted drug that matches their tumor approximately, I think, 2 or 3 months into treatment, and then within 6 months, their cancer became resistant, and they're right back where they started. So we see this in lung cancer too, not as visibly dramatic. So essentially, at the beginning, I had that wagon wheel where there were all the patients in the center, and we parsed them out into the green group, the yellow group, the red group. I think the paradigm now is, instead of a wagon wheel, it's more of a circle or a spiral, because it keeps going around and around, where we would have a patient do a biopsy to obtain some information about the biology of their tumor, use that information to choose a treatment, and then when the treatment no longer worked, we need to go around the cycle again. So we have been doing biopsies on patients with acquired resistance to targeted therapies for 3 or 4 years rather routinely here. I'm sure Dr. Shah will talk about some of the findings that we've had with the ALK patients. But essentially what we've found with the EGFR patients is that there are two basic categories of how patients become resistant to these drugs. One is alteration of the target itself, so in this case EGFR. The EGFR, the most common thing that we see in EGFR-acquired resistance actually is a secondary mutation in EGFR called T790M, which is an acquired resistance mutation. But you could also have amplification of EGFR, basically anything that is going to make it so that the drug, in this diagram, the drug is the triangles. The specific drug can no longer control signaling from that RTK. And then the second major category that we've seen is a bypass track. So on the right-hand side of the slide, you've still got EGFR. It's still being effectively inhibited by the EGFR TKI, but the downstream signaling pathways, basically a detour is created so that they are stimulated by a different RTK, either a mutation in another RTK or amplification. So when we've biopsied lung cancer patients, this was a paper that's now 3 years old, almost 3 years old. This was our initial experience with 37 patients that had acquired resistance to either gefitinib or erlotinib, and then we biopsied them at the time that they were growing, and we compared in a very detailed way their pre-treatment sample with their post-treatment sample. And this was our first pie chart, but maybe we'll go over in more detail an updated pie chart from when we had approximately 100 patients. At this time we have almost 150 patients that we've biopsied and studied in this way. So the results are fairly consistent, and also with what's been published from other groups, which is that T790M is the big player in the room. This makes up 50 to 65% of the resistant cases. And we see T790M mostly alone, but we have seen it in combination with EGFR amplification in a subgroup of patients. So this is the target alteration mechanism of resistance. And then, something that probably our paper from Science Translational Medicine was quoted the most because of this phenomena up in the corner, which I'll talk about next, which is this transformation to small cell lung cancer. This is something that we described and has also been published by some other groups in case report format, that every once in a while you have an EGFR patient who clearly has adenocarcinoma at the time of their diagnosis, but after they've been treated with an EGFR TKI, if you do a repeat biopsy on the resistant cancer, it looks like small cell lung cancer. And these are never smoking patients who you wouldn't expect to get de novo small cell. And in fact, when you study, in our case, when we studied the small cell lung cancer samples, they always have the same original EGFR mutation that the patient started with. So we do believe that this is an evolution of their cancer, not some sort of secondary diagnosis. And in about half of the small cell transformations that we have seen, they also have a PI3 kinase mutation. Then we've also seen some other bypass tracks like PI3 kinase mutation alone, and MET amplification, and BRAF mutation. HER2 amplification has also been described out of the Vanderbilt group. So this is essentially the landscape of where we are with EGFR acquired resistance. So to talk about the small cell for just a few minutes. So this is from some of the original data that we had published when we had 5 patients. Now I believe we have 8 to 10 patients at MGH that have been documented to have this. Again, all the patients clearly have adenocarcinoma at the beginning. You can see in the lower left corner an H&E that looks classic for adenocarcinoma with glands, and that later on the repeat biopsy it looks much different with small blue cells, crush artifact, etc. In addition, the resistant tumors express neuroendocrine markers, which the baseline tumors did not, synaptophysin and chromogranin. But in every case, the genotype that they had originally, the original EGFR mutation, remains. And in a couple of cases now, we've seen the acquired PIK3CA mutation. When these patients have been found, they were treated with a small cell-like chemotherapy that would not typically be given to patients with adenocarcinoma. So here's one example, a patient of Jeff's that had this large axillary lymph node conglomerate, as well as a very painful chest wall mass. And when we found out this was a small cell transformation, she continued on her erlotinib because we didn't want that part of her disease to flare, but carboplatin and etoposide were added, and she had a beautiful response, as you would classically see with a small cell lung cancer patient. One of the postdocs in Jeff's lab, Matt Niederst, has been working a lot on the biology of this phenomenon. So I'm going to show you a couple of slides now that he presented just last week at the AACR triple meeting that was held here in Boston. So we have one particular patient of mine who had this transformation, from whom we learned quite a bit, because for a variety of different reasons, including her own desire to help us understand why this was happening, she went through a number of different biopsies over the course of her disease and went through an autopsy at the time of her death. So this patient had adenocarcinoma with an L858R mutation when she was originally diagnosed. She was on first-line erlotinib for a little over a year, as is completely typical. When she progressed, we did a biopsy, and she had transformed to small cell as well as acquired a PIK3CA mutation. And she was treated with a small cell-like regimen of chemo, and actually in this case radiation as well. And then after about 9 months, when her disease recurred, it was adenocarcinoma. It was outside the field of radiation. It was a pleural effusion, actually. That was adenocarcinoma with L858R, but no PIK3CA. And she had another shorter-lived good response to erlotinib, actually, at that time. This was a little bit before we understood this disease flare, so she had a break from her erlotinib and then went back on it. But from that point forward, when she became resistant the second time, most of the biopsies that we took were small cell lung cancer, and always that was in combination with the PIK3CA mutation. When she died, the vast majority of her disease was small cell with the PIK3CA and EGFR mutation, but she did have one area on the pleura and the diaphragm that was adenocarcinoma that at that point had acquired T790M, different resistance mechanism. And here's a picture which is, I believe, from her diaphragm, where you can literally see, this is from her autopsy, you can literally see this high-grade neuroendocrine carcinoma sort of arising out of the adenocarcinoma. And so we've studied her various biopsies and her autopsy extensively, and again, this is work by Matt Niederst. And you can see that what he has found is essentially that RB is lost in the transformed tumors, and as you're aware, RB loss is kind of a classic feature of de novo small cell, but it's not something that you would typically see in adenocarcinoma or EGFR mutated adenocarcinoma. So from her autopsy, for example, the normal liver and the diaphragm, which was the adenocarcinoma histology, show that RB is preserved, or at least in one allele. But then when you look at the small cell histology samples from the liver and from another part of the lung, you can see that there's bi-allelic loss. And then this is borne out as well in PCR-specific analysis. And then what's at the bottom, these gels, is something a little bit different. These are, I'm sure Jeff will talk about some of our patient-derived cell line models that we are essentially growing from patients that we do a biopsy to try and really understand and dive into the biology of each specific patient that we have in clinic. And so what's shown here is that looking at some of our resistant patients who were biopsied and had another mechanism of resistance like T790M or EMT, that they preserved RB, but that the patients who transformed to small cell lung cancer, as well as some cell lines that are small cell lung cancer cell lines, have all lost RB. So hopefully this will be something you'll be reading about in publication very soon, but very exciting work about the mechanism of what's causing that small cell transformation. So now I want to turn to the most common resistance mutation we see, which is T790M. So we've known for a while that this is the most common resistance mechanism to erlotinib or any of the EGFR TKIs. And clinically, it seems like these patients may have a somewhat better prognosis, that their disease may be a little bit slower growing. I think back in 2005, this is a paper that came out of the Haber lab when Eunice Kwok was a postdoc there, looking at what was then the first in class of the second generation TKIs that this was thought to be very promising. This was going to be the answer to acquired resistance. Essentially, the second generation TKIs are irreversibly binding, so they actually form a covalent bond between the drug and the EGFR protein. And they also have a broader spectrum, hitting not only EGFR, but HER2. And so one of the first drugs, which was a Wyeth drug, HKI-272, it looked very promising in lab-resistant models, including T790M. But unfortunately, what we saw unfold over the next 5 to 7 years was that as these drugs were tried in patients with acquired resistance, we didn't see too much. Both neratinib, afatinib, and dacomitinib, which is the Pfizer second-generation TKI, the response rate was under 10%. And so this was a big disappointment, I think, for the field, a lot of frustration. Why were these studies negative? I think we'll never know for sure, but I think part of it is that when you treated these models in the Petri dish, you could give a lot more drug, and you could really get enough concentration to be able to inhibit those resistant clones. In patients, we're limited, of course, by the tolerability of the drugs, and so the second generation TKIs do tend to cause more rash, more diarrhea than the first generation TKIs, and I think we were unable to really dose patients at a level at which we could expect to see their resistant disease respond. Finally, I think there is a solution coming to this problem with the third generation TKIs. So there are 2 third generation TKIs that are in human studies now. I'm going to talk primarily about CO1686, but there's also an AstraZeneca compound that I think also holds a lot of promise, but I don't have as many slides about. So CO1686 is basically a third generation TKI, which means that it does very selectively inhibit EGFR mutated EGFR mutations with the activating mutation, and it also inhibits T790M, the primary resistance mutation. But the EGFR that it does not hit is the wild type EGFR. So with this drug, even preclinically, when they saw this, they expected there to be very little rash, very little diarrhea, very little of the side effects that come from wild type inhibition. The first in human study is still ongoing, and there's been some change in the formulation. It was initially a free-base drug that was given, and now hydrobromide salt is being dose escalated. But essentially what was presented just a few hours ago in Australia at the World Lung Cancer Meeting is that there's been this is for 56 patients dosed so far, very reasonable side effect profile. The majority of patients actually are having almost no side effects. If anything, it's predominantly grade 1, nausea, a little bit of diarrhea, fatigue. No rash has been seen, and grade 2 and 3 events are very rare. And of the dose, the highest dose that's been given and fully evaluated is 900 milligrams BID of freebase. Of the patients who were T790M, which is really where this drug is thought to be able to work, we've seen a 67% response rate, and 8 of 9 patients have had tumor shrinkage. So this is a big change, I think, at least in early days, from what we saw with the second generation TKIs, and this is a very exciting area of research currently. Here's a patient from Mass General who was heavily pretreated with multiple TKIs in her history, who had a baseline, a lot of tumor. I'll show you some different areas, but here's one particular area in the left lung. She had a PR to treatment. Here's liver met shrinking, and also we've seen CNS activity with this drug, so it probably doesn't project that well, but she had a midbrain lesion that essentially went away on the follow-up scan. So keep your eye on that space for future treatment of T790M. Now, what about the bypass tracks that we talk about? How can we treat patients that have developed that type of resistance? Well, Jeff probably won't go over the initial work that he did when he discovered MET amplification in his lab, but essentially when that, which was the first bypass track that was shown from the lab work, we saw that in order to effectively treat those cancers, you would have to keep the EGFR inhibition as well as add something that covers the bypass track. And we have seen this especially in one proof of principle patient. This is a patient who was on first-line erlotinib, had a great tumor shrinkage after about a year, developed resistance, and you can see the needle going in for the repeat biopsy. And that repeat biopsy demonstrated that this patient had amplification of MET. And we were able, the timing was right, that we were able to get this patient on a clinical trial of a MET inhibitor, which happened to be cabozantinib as well as erlotinib, and the patient had another PR when we combined EGFR and MET. We haven't been able to do this in too many patients, honestly, because the timing just doesn't work out right, that when you find a patient with a bypass track, you happen to have a clinical trial that combines those two agents. And you know, one of our ongoing frustrations about treating acquired resistant cancers is the difficulty that we face just logistically with combining multiple agents, especially if neither agent is yet FDA approved. So I think this is something as a field we have to work on going forward, is how can we get over the sort of systemic hurdles that prevent us from combining drugs earlier in their development. Another successful treatment for acquired resistance in EGFR, a combination of afatinib, second-generation TKI, plus cetuximab, and an EGFR-specific monoclonal antibody. This treatment, unlike the third-generation TKIs I talked about briefly, is fairly toxic. I think it's going to be a little bit difficult to take this treatment forward into the clinic, but there has been about a 35% response rate, which is, this was really the first treatment to work in acquired resistance for EGFR, and the PFS among the responders is 4.7 months. And then we've also seen that some of the HSP90 inhibitors, in particular the Novartis compound AUY922, has had some responses in EGFR patients. And again, this is data that was just presented this weekend in Australia, showing a response rate of 36% among EGFR mutants treated with this HSP90 inhibitor. So in summary, I think the genotype-directed paradigm has really completely changed the landscape of how we treat EGFR, how we treat lung cancer patients, and EGFR was kind of the first kid on the block. We still have two major obstacles to understand how to improve the response for all patients, in other words, how to decrease intrinsic resistance, and also what to do with acquired resistance. We will be looking, as far as what we're doing here at MGH, we are going to be looking at how we can modulate initial therapy to try and overcome intrinsic resistance, and we have a fellow, Zosia Piotrowska, who's working on a trial that will personalize treatment within EGFR patients based on BIM levels, and then also moving some of these newer third-generation drugs into the frontline setting, I think will may improve outcomes. We have been learning that small cell lung cancer transformation is mediated by loss of RB, and there may be actually opportunities to try and treat that with targeted therapies as well, as we further understand this phenomena. I think T790M is actually going to be quite treatable with third-generation TKIs, and as the data on CO1686 and AZD9291 matures, we'll learn more about that. But we need more effective strategies for non-T790M mediated resistance. So that was essentially what I wanted to say. I wanted to thank my collaborators, in particular Jeff and Alice, who are both here, and John Ifredi and Mari Mino-Knudsen, who we work with very closely in pathology. Thanks.
So you said a couple of times that the small cell transformation was mediated by loss of RB. Is that as distinguished from some alternative hypotheses where this is a qualitatively different type of lung adeno that is from a different precursor cell, that it was always there? Or put another way, if you take an EGFR mutant adeno lung cancer cell line and disrupt RB, does it transform into small cell?
Yeah, so that's a great question. And again, I'm not sure how much of this is in the public domain yet, but we have seen that it looks like what you said, the second thing about taking an adenocarcinoma cell line and disrupting RB is not sufficient to cause it to transform into a small cell lung cancer, but we think it is necessary. I don't know, Jeff, if you want to comment as well. Marty, can you give Jeff the microphone? Raise your hand, Jeff.
So the only thing we see is just a really remarkable correlation that in all the resistant cancers you don't see loss of RB except in the subset that becomes small cell. But loss of RB in and of itself is not sufficient. In the laboratory models we've looked at to cause small cell. I will say from Lecia's work that I don't think the small cell that exists when the cancer becomes resistant was actually there when you started treatment, because a good number of these tumors actually explode like small cell does. And a lot of these patients go into remission for several years and all of a sudden they explode. So the precursor of that small cell may have been there to begin with, but yeah, but that actual cell we see it. Yeah, we don't. Oh, so the question was whether or not, even before treatment, were these cancers, were the cell of transformation a more stemmish-like cell, and so they had the potential to become small cell more readily, and they would be distinguishable by expression analyses. We don't know the answer to that. We don't know the answer. We haven't done those experiments or those assessments. I had a question though for Lecia. Right. You know, the dabrafenib resistant, not dabrafenib, the vemurafenib resistant slide you showed from Levi's group with the melanoma that goes away and comes back. What's remarkable to me is that every one of those resistant lesions, and there's got to be like 50 of them, exactly is where the previous cancer was, and they all come back at exactly the same time. And I don't know if that's a unique case. I was wondering, from the experience in EGFR, do you see that, or is it more random that sometimes just one lesion will come back, or do they all tend to come back at the same time, like that very dramatic situation?
I would say it's very heterogeneous. There are some patients, I don't know if you remember the slide that I showed with the chemotherapy continuation, where there was like sort of a larger lesion and a smaller lesion, and it shrunk, and it did come back, those exact same two lesions on that slide. But I would say that only happens in maybe 40% of cases. We've actually looked at this a little bit with one of our radiation colleagues to try and see if at the time of maximum tumor shrinkage, if you radiate sort of the minimal residual disease that's left, if you could delay the onset of resistance or of clinical decline. So we're going to be looking at some of that prospectively, but there are patients who will come up with a new metastatic site, sometimes in the brain, for example, that they didn't have at the time of diagnosis. And sometimes it will be one spot and then more, instead of all at once like that melanoma patients seem to have.
One more question, Mike.
I'd just like to come back to the small cell. First of all, very nice talk and review of the EGFR in lung cancer. So the paradigm that I've always worked with was that you only need one hit in the P16 CDK4 RB pathway. And so you're describing loss of RB in the small cell. If you look at the patient's samples in their adenocarcinoma, do you see deletion of P16 or methylation or some other mechanism of loss or amplification of CDK4 or cyclin D1?
So I don't know if we've looked at all of those different factors. We have done whole genome sequencing in some of the samples pre and post, and we see that even when the samples are still adenocarcinoma in appearance, that they do have loss of one allele of RB. It's almost like they're sort of primed to make that jump. Do you want to add anything else, Jeff? Or?
No, but I mean, I found that in the same, as Lecia showed, in the same patient, and this goes a little bit to your question too, in the same patient, you have some lesions that are adeno with the T790M and some that are small cells. So, you know, it really is not a fait accompli that is a tumor before treatment was going to become small cell. And there aren't any predictive biomarkers that we've had for which ones will become small cell readily.
what is the status of the other members of that pathway, and do you get something in addition by losing RB, or is it that you don't have loss of P16 and you lose RB
You
downstream?
mean in the small cells?
In the adenocarcinomas and in the small cells, what is the status of P16, CDK4?
Yeah, so P16 in general has been intact in both the small cells and the adenos in the tumor specimens. And that has not been a distinguishing factor. We're actually looking more at notch signaling, which seems to be very differentially activated in the small cells and non-small cells, but it doesn't seem that that pathway, P16, has to be inactivated in general, which might make sense to some extent because you have the hyperactivation of cyclin D, CDK4/6 activity because the EGFR mutation itself hyperactivates TORC1 signaling, which regulates cyclin D levels. So you may not have a genetic event that's normally activating that pathway, but the signaling itself probably stimulates that pathway quite a bit.